Hey, Mr. YS here, and this is the last bit to go through on digestion. So let's have a look. This is a kind of overall statement, and it does kind of blend into the next bit. But what is the function of the small intestine, and what aids this, and what features does it have? So the small intestine, we know that it is this tube that goes backwards and forwards, and that it has a cross section, and it has many sections in. But thinking about the main function, we're talking about the mucosa layer and what's happening in the mucosa, which has villi. And villi are these finger-like structures, and obviously they go round in a circle. So this is extending all the way round in 3D. So we've got these finger-like villi, and the whole point of the small intestine is absorption. It is going to absorb things in. Now, what kind of things is it going to absorb? It can only absorb things once they have got down to the monomer level. It can only absorb monomers. So it can't absorb polymers. So the whole point of digestion is to get to this point where we have monomers and they can then be absorbed into the blood vessels, which are all connected underneath here and taken away. And also we've got these lacteals, this lymphatic system, which I'll draw as kind of yellow, the fat. So we need to get monomers into them, into both the blood vessels and into the lymphatic system to then transport it around the body. It will go back to the heart and get pumped out. So here's one monomer over here. Can you guess what it is? That is glucose. So we get the digestion of sugars, the production of sugars from the digestion of carbohydrates. We could be breaking down, and I'm just going to draw some different shapes here. This is not really what they look like, but we have long chains of amino acids, which are in either, if they're sort of chains of them, we could call it a polypeptide. And by this point, we've probably started to chop them it into shorter polypeptides. And then eventually we want to get amino acids, and then the amino acids can go through and into the blood just like sugar molecules. Okay, so what's the other thing then? The other thing would be our fats, and fats are, the most common type is called a triglyceride. And so this was made out of glycerol, which is a 3-carbon molecule, and then we had 3 fatty acid tails. So these triglycerides, they would be linked together, so it'd all be together like that. And this can be broken down as well. We can get enzymes coming and chopping them. And so what they get chopped into to start with is, and I'll draw it as the zigzag structure. Remember, this was our carbon chain, which could be a saturated or unsaturated fat, depending on whether it's got double bonds there. So we can break it down into, we can see there's one chain. And so because there's one chain, then this is a mono for one glyceride, because we've got the glycerol with its 3 carbons. This would be 1, 2, 3 carbons which are there. So this is 3 carbons, but it's a monoglyceride because we've got one fatty acid chain. And then what have we got left? Well, we've got 2 fatty acids. So we can say that fats are digested into monoglycerides and fatty acids. And the fatty acids can go through and into the lacteal, and the monoglycerides can also get through and then get into the lacteal. So that's the whole purpose. I remember, of course, the other purpose is we've got kind of contraction of the circular muscles to make the ring smaller, to push food through, and peristalsis, so its function is also to be mixing. Okay, so let's have a look at that as a whole. Well, overall, we can say it is where food substances, which are nutrients, are, well, what happens to them? They're absorbed into the bloodstream by different processes, and we'll have a look. We've got diffusion, active transport, and facilitated diffusion. Okay, what else happens? Well, we have monomers, we talked about that. Like fructose. We also talked about amino acids, monoglycerides and fatty acids, and also glycerol if that's been broken down, if it's been removed. And we said it's lined with smooth muscle to allow the mixing and moving of digestive food products through peristalsis. Next, let's consider what the features of a villus are. So here we are, we're zooming in on a villus. Now, one thing to make sure that we are really clear on is that there is a difference between a villus and the cells of the villus. So here's a whole villus, and a whole villus is made up of cells. These are epithelial, because they are lining cells. Epithelial cells. So there are many, many cells, and then it's also made up of, we've got this rich capillary network, which is bringing blood in and taking it away. In the middle, we talked about our lacteal, which is where fats are going to get absorbed. And if we zoomed in, so this is over here, this is us zooming in on one of these cells over here. If we zoomed in on the cell, it would have these kind of finger-like projections. So this would be the cell, many of those making up the wall. But if we zoom in, it's got what could be called a brush border, because it kind of looks like the bristles of a brush. And these little things here, they are microvilli. So tiny little structures which again are going to have this role in like further increasing the surface area. So surface area for absorption. So let's just have a think about what these things do then. So in turn, and we'll come to a way to try and remember some of them. If we're talking about the features of the villus, well, the villus has, because it's the whole thing, the whole villus, if we're talking about the whole villus, well, yeah, it has a rich capillary network. So what does that do? That is about providing a flow of blood. And that means that when things move into the blood, they then get pumped round and get pumped away. So if we were going to say that there was some glucose in the gut and it could get absorbed in, well, as soon as that glucose gets absorbed in, it now moves away. Because otherwise, if we didn't have this good blood flow, the glucose would move in, and then we wouldn't have a concentration gradient, so it wouldn't happen fast. So the rich capillary network maintains this difference in concentration, which we call the concentration gradient. So it's got a good blood flow. What else? Well, we can see over here that it's only a single layer of cells. So there is a single layer of epithelial cells. Now what's that going to do? Because at IB we have to say what does each of these things do. By having only a single layer rather than multiple layers, we reduce the diffusion distance. And then we're going to go back to a stock phrase here. So if we decrease the distance for diffusion, what's going to happen? We are going to increase the rate of absorption. So we're going to go back to that as being a kind of phrase that we can use for lots of things. Okay, what else about the villus then? Well, it contains a lacteal, and that was for the absorption of monoglycerides and fatty acids. So that is kind of a feature just by itself. Okay, so those are the features of the whole villus, and where people come unstuck is that they then talk about the features of the cells of the villus. So now we're going to talk about, well, what are the features of these individual cells of the villus? So where we see this word cells, we're not talking about those previous things. We're now talking about what would a cell be like? What kind of features would these cells have to mean that they are good at being the lining cells? So just stop and think about what kind of things would you have in this in terms of cell structure. We talked about, well, one feature is definitely the microvilli, but what other features would they have? Okay, well, they're involved in a lot of active transport. They're using a lot of ATP. So what kind of things will they have? Well, they're going to have a lot of mitochondria, which are the places where we're producing ATP for use in active transport. So they have a lot of mitochondria. What else is a feature of these cells? Well, they are close together, and the gut doesn't want to let things through in between. That would be bad. This is called leaky gut. If things are going through those cells. So there are special proteins which kind of stitch. There's a stitching between these. So what do I mean by that? I mean there are proteins that, like stitches, they kind of hold these together. And these are called tight junctions. Tight junctions so that we don't get food or other things leaking through. Okay, the other thing that we talked about was they've got these microvilli, and they are going to increase the surface area. You can't just put SA, I've just written that there as SA. But surface area, and going back to that phrase, for faster absorption of monomers. Okay, what else? Well, the other thing is that in between, or part making up these cells, other than the cells which do the absorption, there are also cells called goblet cells. And what they do is they release mucus. And that mucus helps to lubricate, to help the food move through. So you could try and use the Mrs. Limpt mnemonic. And what's Mrs. Limpt? We're just going to erase all of that. So Mrs. Limpt was a way of trying to remember these different things. So let's go down through. M, so M for microvilli, increasing the surface area for faster absorption. R for the rich capillary networks to help maintain the concentration gradient. We've got S for a single epithelial layer to have a minimal diffusion distance for, again, faster absorption of, we could add in monomers. Lacteals to absorb the lipids. This, I think we can ignore. Intestinal crypt located that release juices. Well, I think that you don't really need that. And then we talked about M. M we could just have for mitochondria, but they also have membrane proteins. The other thing that isn't mentioned there is that there are enzymes. So these proteins are enzymes like maltase. So maltase is stuck in the epithelial cells. It's a membrane-bound protein, and maltase can break down maltose into glucose. Okay, so lots of mitochondria because we've got lots of active transport going on. What is pancreatic juice? Where is it released from? What does it contain? What's its function? Well, I mean, it's released from the pancreas. I think that's going to be quite obvious. And here we have all three enzymes. And in fact, there's a range. 3 enzymes because we are going to digest proteins or polypeptides. We're also going to digest carbohydrates. And we're going to digest fats. So the 3 things, we're going to have a lipase, that's the fats, lipases, so we've got pancreatic lipase, which is going to help in the digestion. And we talked about the idea of triglycerides. Which were these, the glycerol with 3 fatty acid tails, triglycerides down into monoglycerides and fatty acids. Next, let's think about the proteins. Well, these proteins have already been started to digest by the protease pepsin in the stomach. So these are actually polypeptides. So we're talking about what the substrate is, the thing that gets digested, and we're talking about what the products are for these enzymes. So for pancreatic protease, which is going to help digest polypeptides into shorter polypeptides. So we haven't yet got to the amino acids, so it's just going to be creating little cuts in these chains to make them shorter. And the name of this is trypsin. So pepsin was in our stomach and trypsin comes from the pancreas. So it's going to do polypeptides into shorter polypeptides. And finally, our carbohydrates. Well, we've got an amylase, pancreatic amylase, which is going to digest our larger polysaccharides and it's going to digest them down into disaccharides. So remembering that polysaccharides were lots of glucose linked together, maybe to make something like amylose or amylopectin. And those two things, amylose and amylopectin, make up starch. So we then get down to disaccharides, we would get down to the level of maltose, for instance. And it depends on what the polysaccharide was. Okay, so it's producing all those things, and we could also talk about, well, what kind of cells are producing these? Well, if they're secreting into a duct and it's not into the blood, then it's outside the blood, so we could say that they are exocrine cells. Exocrine cells here are producing these enzymes to secrete in here. The other thing that we mentioned before in the other video is that they're making endopeptidases, which cut in chains down to amino acids, and also exopeptidase, which cut at the end of a chain. So an exo cuts right at the end, and that means it will cut off the last one, and an endo cuts within, and eventually now we're going to go right down to an amino acid. Alright, so let's have a look at that as a whole thing from the flashcards. Well, the pancreatic juice is secreted, that should say. It's secreted, or it's released by exocrine cells of the pancreas, which contain enzymes amylase, lipase, and proteases. And we talked about what would happen. Starch would be digested, or amylose and amylopectin would get digested down into disaccharides. And lipases to digest triglycerides, and the other thing that they also digest from our cell membranes is phospholipids. And then the proteases digesting proteins into shorter polypeptides, and then finally our endo and exopeptidases to make something which will digest the shorter polypeptides into amino acids. So once we got down to the level of a monomer, and we talked about those different monomers, e.g. glucose, amino acids, and monoglycerides and fatty acids, how are these going to be absorbed? What's the method of absorption for them? So just stop and have a think. How would those get in based on what you know on this about the cell membrane? Okay, firstly, glucose. It's large and polar, or large enough that it doesn't fit through the membrane. So it's going to need to have channels to go through. So the good guesses would be facilitated diffusion, which it is actually. But because we use ATP to pump, we can call it overall active transport. So there's active transport of glucose. Amino acids, they are also large enough and polar enough that they need to be actively transported. Monoglycerides and fatty acids, because they are large but non-polar, they can actually diffuse in. So they diffuse in and then they get packaged up into a lipoprotein, which can then carry it in the bloodstream when it eventually gets there. So here's our review. Different methods. We get simple absorption, and by that we mean diffusion, really. Diffusion of fatty acids and monoglycerides. There is some diffusion of amino acids, but we can also say it's active transport. It depends on the gradients involved. We get facilitated diffusion of glucose. So that's the high level statement, because really it's co-transport with sodium ions. We use pumps to pump out sodium and then co-transport in glucose. But we can say that that's active transport. and we get diffusion of fatty acids, and then exocytosis via this lipoprotein that we stick the fats into the lacteal. Okay, this next one is a pretty straightforward one. So what does the large intestine do? It absorbs the remaining water. It also absorbs dissolved minerals, and it has, inside it, it has lots of bacteria. So it's alive with loads of different bacteria, and the bacteria help to make the vitamin K. And they also help to break down nutrients and help you absorb the rest. There is some nutrient absorption in there. So it's the final absorption of the remaining water, dissolved minerals, and helping in vitamin K absorption. So what's the difference then between absorption, taking things in, and assimilation? What's the difference? See if you can describe. Well, absorption is only the movement of a fluid or dissolved substances across a membrane, and we're thinking about there really across a membrane and into the blood. Whereas assimilation is when it becomes a part of you. So you take them up, cells take up things and use them. Okay, moving down to starch digestion. So how is starch digested? Well, we talked a little bit about it before, being made up of two different things, amylose and amylopectin. And we also talked about the enzyme. So it's made up of amylose and amylopectin. Quick review of those things, because there's amylose, which is a straight chain, because it's carbon 1 to carbon 4. Now, when we remember drawing glucose, carbon 1 was over there and carbon 4 was there. So that gives us these straight chains in amylose. Carbon 1, 4. But carbon 6 is at the top. So when we're bonding to carbon 6, then we get this branching. We get branching happening. So amylose was our straight chain, amylopectin was branched. But both of them are digested by amylase. It will start to break down into maltose. And then finally, the maltase enzyme will digest maltose into glucose. And it's in the membranes of the villi, so it's an immobilized enzyme. And then eventually it's going to be transported into the blood and go to the liver, and then the liver might start storing it. Okay, final bit. So what can we use to model the small intestine? Well, the small intestine has a cell membrane that is partially permeable. It will not allow things which are too large to get through. And the way it does that is these gaps that I'm creating there, they're not real gaps. They're actually if we're thinking about our phospholipid bilayer, they are the channels. So these are channel proteins, because we talked about facilitated diffusion. Pump proteins, it's the membrane being partially permeable. And so the way that we can mimic that, and this is actually used in dialysis for kidney failure, we use something called dialysis tubing or visking tubing. And the visking tubing has these small holes which will allow glucose through and urea through and water and salts. But they won't allow things like maybe a red blood cell. So the blood cells can't get through. The white blood cells can't get through. And so what they do, how this models it, is that large things as well, starch, large things cannot get through the gaps. And then we can show that only when we digest into glucose, if we add, or in fact maltose, if we added some amylase here, then these could diffuse through. So the gap is small enough that glucose could get through, and we would get glucose over here. So how could we test those sides? If this was outside and this was inside the Visking tubing, how could we test for starch and how could we test for glucose to see where it had gone? And just stop and pause and think about that. Okay, so this is the situation we're thinking about. We've got a beaker where we have some Visking tubing which has these pores in. So if we put in there starch, then how could we test for it? Well, we could test using iodine, which would go blue-black. Now, if we tested the outside, would it go blue-black? Well, no, because starch is too big. So blue-black on the outside, and here in the beaker on the outside, it would still be the orange brown for iodine, because the starch can't get through. Starch is the long chains of glucose, and that, if we put it in there, it's not going to fit. Then we can show that if we add an enzyme like amylase, it's going to start cutting those. So if we put the amylase in the Visking tubing, then we're going to have glucose being made. Sorry, not glucose, we're going to get maltose, we're going to get these disaccharides. And that's going to be able to move through. So initially, if we tested the inside with starch, it would go blue-black, but the other test that we can do is for sugars. The test for sugars is called Benedict's. And it goes from blue, because it's copper sulfate solution, blue, it turns brick red. So at the start, would the test be positive? Well, no, because there's only starch there. But afterwards, if we do the test again, well, yes, we're going to have sugars there, and yes, we're going to have sugars there, but we still won't have starch. So our starch hasn't moved through. We will always get a negative test outside. It's not going to go blue-black. The inside would still probably go blue-black until the very end, until all of the starch had been digested. But once we add the amylase, now we're going to get a positive test for Benedict's. Okay, that's it. Thanks, folks.